Hello, everybody, and welcome to today's class on global clustering. I'm really excited for today's class because whenever you cluster your data, you get to kind of know how it all relates to each other and look for genes you know and see if they're making sense. And so we've been doing all of this work to get to this point where we're actually going to start to see the relationship between these 400 or so DNA binding proteins that we've been analyzing by Chip-seq. And so what you can think of hierarchical clustering as basically a family tree, if you will, that what samples are more similar to each other. And the way we're going to look at this, because we're looking at chip data, is whether a DNA binding protein likes to bind to the same promoters as another DNA binding protein. And so we made a profile. I'm going to go back because we've been doing a lot of stuff, and I think it's good to take a step back and go over one of the functions or two of the functions that we were running and see what they did for us and how powerful that actually was to give us, in the end, we got a matrix where every column is a promoter in the human genome and every row is a DNA binding protein. And what we will say is if there's a 1, if there was a peak that intersected that promoter, and a 0 if it didn't. So we'll have a string of about 30,000 ones and zeros for each DNA binding protein of whether they did or did not bind a given promoter. And then we'll use clustering to find out which of those 1s and 0 patterns are more similar to each other. And then eventually it will cluster to the point we find which ones are very different from each other. So let's get started with our 438 DBPs that we filtered by having at least 250 peaks. I've already pre-run everything today because a few of these are going to take several minutes to run. But feel free to just pause the video and wait until they finish for you. So I've outlined sort of what I just said there. Here's the libraries we'll need for today. There's a new one, ggdendro. This will draw that family tree for us in the end in ggplot. So what I wanted to go over real quick is something we did in 10. And I know that lecture was very long and had a lot of code in it. So this class will keep iterating back over things. So don't feel worried if you were a little lost there. And so one thing we've been doing is we loaded in GenCode. We made this GenCode GR object, which is really just loading in the GenCode annotations of the genome. And as we saw in 10, the 10 RMD file, that there's all kinds of features in there, number of exons, promoters, gene bodies, et cetera. And so we made this get promoter region function, which we discussed briefly and is very simple. We actually did this same indexing. But now moving forward, we can always just use get promoter regions to do that, where we have already set it to 3 kb upstream and downstream of the transcriptional start site of an annotated promoter. And so what this will do is it'll go and you'll give it the parameters and it'll return your promoters. So then we had a file of promoters. And then we printed those promoters out. Again, you don't have to do this today. We already have these in our data file from the 10 RMD that we made a GTF file of all the link RNA and mRNA promoters, so all the promoters. And then we exported that to this GTF. And we're going to want to grab that today. So in case you're wondering why we did that, that's why, is we're going to need that moving forward. We also made 2 separate files, one where we made long non-coding RNA promoters alone, and one where we made mRNA promoters. Okay, great. So we have 3 different types of data, and we can actually cluster all the promoters, just the long non-coding RNA promoters, or just mRNA promoters. And we'll do all 3 today. Okay, so this function we used is a bit bulky. It was in the markdown. And it was in 10 and 11, we talked about this count peaks per feature function here. And it's in our util folder here in the intersect R functions if you want to go look at it in its raw form, but it's just copied and pasted here. So we also went over some of the function annotation, which really just differs by this little tick mark. And so we have a description where this is going to take a subset of biotypes of promoters, and it's going to count the number of times your Chip-seq peak file overlaps these features. And so you can go back over this again. I know it seemed like a lot in the previous class, but now that you've had some time in ggplot and other things, I think you'll understand this a little bit better. So please go through this and we'll discuss any questions you have in class. But I want to point out a couple things. What this will do is it will return a type. And we either want a count is what we were using in that data frame. So it just counts the number of times a given DNA binding protein binds a given promoter, or we have occurrence. And that's what we're going to use today is the occurrence matrix. We did export this in the 10 lecture. And so what we want for today's is the occurrence, which is a matrix that where every column is the promoter and every row is the DNA binding protein. But it's in a matrix format and not a data frame format, which makes it have the ability for each row in the matrix to be correlated to each other. And we printed this out as this peak matrix as an occurrence matrix in the lecture 10 in results folder. And that's right here, this link RNA, mRNA, promoter peak occurrence matrix .tsv. You'll notice the TSV is different than the CSV, the data frame that we just used for the 11.rmd in ggplot introduction. Okay, so we're going to grab those things we've already made, but I just wanted to go through where we made them and how we made them, and feel free to go through this and we can discuss more in class. And so lastly, I guess the last thing we did that you don't have to do today is we printed this out as the promoter peak occurrence matrix TSV, and we can see it here in our results folder. Okay, so now let's go grab those files that we made, since we already have them handy and we'll use them again. So you'll want to run this line here at around 157 as of this recording. Shouldn't change much, but just in case. Where we're going to call an object promoters, and we're going to call up our track layer, which will be a granges, in an import this long non-coding mRNA promoters. And we're going to load in our peak occurrence matrix, which is going to have all, it's going to have all 460 to start with DNA binding proteins. We did filter the CS.CSV data frame by 250 and got down to 448 DNA binding proteins that had at least 250 peaks. So we'll do that again here today, and I want to just discuss that quickly, but go ahead and load these in, and in your environment, you should have a promoters variable now, or object, and a peak occurrence matrix here for now. Okay, so now to filter this peak occurrence matrix, we're going to make another object called peak occurrence matrix. Maybe should have named it filtered, but it's fine. And what we're going to do is index into peak occurrence matrix. And I wanted to go over this because you could think that you could do this in front of this, but because it's a matrix, we have to index into it. If it was a data frame, we could do this a different way. The details don't really matter. But again, this is where we're going to index and run a function inside of an index. So we're going to go into our peak occurrence matrix object, and we're going to run the base R row sums function. So what that's going to do is take the row and add up all those zeros and ones, and hopefully it'll be more than 250. And so we'll do row sums on peak occurrence matrix and make sure that they're greater than 250. And this comma with nothing means that because we did row sums, we still want to keep all of our columns. So we're just saying filter all the rows, because rows come first, that are greater than 250 when summed, and leave all the columns the way they were. Okay, so go ahead and load that in, and you'll have your peak occurrence matrix that will have gone from 460 now to 448 observations here. Okay, good. And so now we're going to make another object, which is really going to hold all of the correlations. So now what's going to happen is each row is going to be correlated to every other row. And so let's say 2 rows were identical. Their correlation would be 1. Now, if they were exactly opposite, their correlation would be -1. And we'll discuss that more in some of the statistics lectures that are coming soon. But for now, just think it's a similarity of how similar are the ones and zeros. And that the business end of that is going to be this function hclust, which I think is worth giving a look here. And so you can see it's hierarchical clustering, it's base R. Again, another great aspect of R is all the statistics that are built in here. All kinds of different methods you can play with. I suggest trying to change some of these parameters and see if anything changes if you're interested. But really, we are kind of limited to one type of clustering, and that's binary. And that's shown here that in method, there's the method arguments here, and we can add binary. So hclust is going to do all the correlations, and it's going to find the distance between them. And the distance is going to find the correlations, and then this is going to cluster them to how similar those correlations are. It's going to make a matrix of how similar the correlation values are. Oh, it's going to make a matrix of the correlation values. And then we're going to run that out of our peak occurrence matrix, and we need to tell it the method is binary. In RNA-seq, when we're going to do some clustering there, that's a continuous variable where you don't just have ones and zeros, you have 1, 50, 70, 6,000. So it's more of a continuous function, and you want to think differently about correlations between binary and continuous. So we're going to set the proper method to binary here, and I believe I have been, yes, I do. So go ahead and run this. This might take a few minutes. But go ahead and run this guy until you have the bin here object. Okay. And this is just to take a look at this quickly, see what we got inside there. And what we can see is our 448 different DNA binding proteins, just sort of showing the order in which they clustered together. This is very hard to gain any information from because we don't have the branches to know how close or far away they were from each other. So let's do that. And that's where we're going to call up ggdendro, which is going to plot the tree branches for us. And because you guys are now getting good with ggplot, I want you to look through here. We talked about scale x continuous. And what we've done today is just added some new indexing to it. So take a look at that, and we'll go through it. But it really is following the same fundamentals as we did in 11 with the introduction to ggplot. So ggdendro needs its own parameters, so you can simply manual ggdendro. And did I spell that wrong? Yes, ggdendro. Yeah, let's do ggdendrogram. Oh, that gives us the base code. Oh, that's kind of cool. What we want is ggdendro. There we go. Okay. And now we can see what it's going to do for us is create dendrograms and tree diagrams using ggplot2. So really, it's like doing ggplot, but saying ggdendro instead. Theme. We have a theme here. This is just all because we have so much data. We wanted to pre-cook it so that it's actually going to be readable and interpretable. The theme here is being set. We didn't just do the paper white like we did last time. We added some elements here. Don't worry if this doesn't all make sense. What I'm hoping you can see is how we added the layers of the graphic, the grammar of graphics to build this figure. And so really the only difference we're doing here is doing ggdendro instead of ggplot. So let's run this and see what we get. It will take a minute. Okay, actually that was quite fast and totally unreadable. Don't worry, we're gonna fix that in a second. And you could export this or zoom to get a better picture of it, but really you can't read much yet. But what I do want to point out is that what's going to happen is this program is going to find all the correlations, and we can see that the longer the length of this branch, the more distant they are from this origin. So this would be the origin of our plot. You can think of it as a family tree, and then we can see that it's a long ways over to this group, and it's a long ways over to this group. So that's already a good sign that we have texture in our data. In fact, it looks like we have 3 distinct groups, one down here, one sort of in the middle, and one over here. And what we can see, because of its longer name, is this is POL2 phospho S5. I have all of this in the markdown in the RMD, so don't worry, or just ignore me for now. And POL2 phospho S2. These are very different forms of the polymerase, and they should not be clustered together. And so we'll take a look at them at the end and just see if this makes any sense. But this is kind of fun because we're starting to see our data all together for the first time, and we're going to get to see if it makes sense in just a couple minutes. Okay, so we've clustered all promoters, and let's save this here. I've already done that and actually drawn them to my desktop so we can look at them at the end of this. So go ahead and save those in wherever you want. What our structure is right now is we're in this class, since we're going to be doing Chip-seq and RNA-seq and other types of seqs, we're making a folder for each type of seq. So now that we're really doing Chip-seq stuff officially, getting results, and actually a figure, if you will, this could be a supplemental figure. So we're going to put this in Chip-seq figures, all promoters clustered. This height and width, don't worry about it too much. We're making a PDF with ggsave, but because there's so many DNA binding proteins, we need the height to be really tall, but the width doesn't need to be that wide, because really we can shrink these bars down and still get the same kind of texture from it. Okay, so now let's just do long non-coding RNAs and mRNAs separately. Again, we made these and just import them real quick with our track layer import so it maintains the ranges. Same for mRNA. And then we are going to do the same as we just did above, where we make a long non-coding RNA peak occurrence, which is the peak occurrence matrix that we've just plotted. But now we're going to say, okay, we want all the rows because we want all our DNA binding proteins. But we're going to now ask for only the gene IDs of the promoters that are long non-coding RNAs. And that's the main juice of this here, is we filter the matrix. We want to keep the total matrix, and we're just filtering it out to the long non-coding RNAs, and then we would do the same thing for mRNAs. But sometimes it looks weird to see this empty thing and a comma, and that's just saying keep the rows and then reduce these columns by this filter. Keep these rows and reduce these by this. Okay, so let's go ahead and run this. And I believe I already have that. Yes, that might take a minute. Same for this. Run that, and then we'll plot it here. And let's see what that looks like. Shouldn't take too long. There it goes. Okay, so we can see there's a little bit of a difference here. These POL2s are a little closer to each other, but my guess is they're still on either side of this main branch division here. And then go ahead and save it as a PDF. Be easier to look at when we're done here in about 1 second. And just do the same thing for mRNAs. Filter out the matrix, mRNAs, make the distance measurements between each row, and then cluster them, which is giving the information about these branches. Then ggdendro will understand that, and let's run that just to see that's working. Cool. All right, so now we can see again these separations. That's good. We don't want them right on top of each other, that's for sure. Okay, so now save that. And I use CyberDuck, but you can use whatever you want to to go and grab these from your server, or if you've just done this on your computer, you'll have them. But they'll be in this Chip-seq folder and figures. I made these yesterday. They're on my desktop to save some time. You could just simply drag these down to your desktop, and then you can look at them on your computer screen where we can get more space. And let's do that real quick. Where these files are here. I'm just going to do the total for today. You can look at the long non-coding RNAs versus mRNAs later, but now you can see we can actually get somewhere here by scrolling and making it bigger, zooming out if we wanted, that it's a little more readable than what we were seeing in RStudio. But look at all this data. This is incredible. And so let's zoom in down here where we have this pol2 phospho2, which is this sort of polymerase running along the gene. And interestingly enough, we see that the other pol2 subunit, which was a completely independent chip, independent of the phosphorylation state, is right here with it. So that's a good sign. And this is why I was excited for today, because we actually didn't know if any of this data made any sense. Another thing you can sort of pick out quickly is this family of the same type of gene, ZNE here, and all the replicates. Remember, we made consensus peaks from individual biological replicates, but now these are different IPs of different family members, and we still see that they are correlated to each other, and that's good. And when we look up here, what we see is this phospho5, which is proximal to promoters. So the phospho2 means that the polymerase is elongating, it's making a transcript, and all is good. And what's interesting is that you'd expect an active promoter to have this histone modification, where histone H3, lysine 27 is acetylated. We'll get into this stuff much more down the road. But these are very euchromatic or very active marks, all shown clustered together. Again, our data is making sense. That's great. More ZNF figure clusters here, these other ZNEs. I wanted to look for, so you can search this PDF. So K9 tri here is also near EZH2. So both of these are heterochromatic proteins that are the opposite of active. And so it's good that they're clustering far away from our active thing. And I could probably go through this all day long. It's so fun to look here again, just so you can see it right away. One cut is some transcription factor that has two different family members. Both of these had replicates. The consensus peaks were merged for each of these separately, and that we can see them clustering together is really good. And interestingly, CTCF is an important protein. I guess you can see why I was excited today is to see what our data finally looks like as a whole. So take some time, go ahead and play around with that, and pick out some favorite proteins, Google them, and see if you find something interesting or some cluster that of proteins you don't know. Google a couple of them and see if they have some related function. So I'm very excited. Our data is not to have been wasted. I actually didn't know that until now or yesterday. So this is good news, and we have a great data set to move forward with. So we will be moving forward soon with metaplots of what are the, instead of looking at these general correlations, what is the promoter, where is it binding on the promoter? Is it binding in the center of the promoter, on the edges of the promoter? And we'll go through that as our next step. But for now, we know we've got about a lot of DNA binding proteins to get to study. Okay, until then, code light, and thank you.